Hello everyone, today we are going to talk about the next set of mutations, the ones that cause all sorts of problems, and that particularly is the frame shift mutation. So frame shift mutations, these are the ones that are caused by adding or removing nucleic bases. This causes a change in the codon frame. These frame shifts might still be categorized as silent or missense or nonsense, depending on how they alter the codon, but they are almost always bad, and they are not very often silent. So this is the case where if you perform an insertion where extra nucleotides are added to the sequence, then you will change the codon frame. And what I mean by changing the codon frame is that if you have a sequence of bases like this, let's start with our AUG, CCC, GGG, and if I go and I make an insertion into this code, right? So let's make a duplication of that. Just put an A there. Even one base, just one base, is going to be a huge problem because the frame, the codon frame, used to be AUG, CCC, GGG. But now your new codon frame is AUG, CAC, CGG. So the frame has been changed. The game is no longer, the game's changed, right? Previously in our point mutations, we were replacing a single nucleotide and that, or maybe even a couple of nucleotides, but it did not change the overall frame. Notice that if we extended this a little bit more, so let's put like U, we would now see that every single codon would be altered from this point on, right? So this would be as right after this mutation, every single codon is different. Changed all codons because of the frame shift. This is the frame shift. It is shifted to the, well, I don't know if we think about left or right, but it's shifted, right? All of the nucleotides, I guess, are shifted to the right by one place. Now, of course, the same effect would happen if you did a deletion. So maybe let's just copy this little frame we made here. Duplicate that, and let's put that down here. If we can somehow, let's set that all up, and then get in here. Okay, clean that up. So if we made a deletion now, let's delete right around that same position. Notice that we would have to start moving things over to fill out that spot because codons are read in sequences of 3 no matter what. There's no way to control for that set of 3. And so what we end up having is we actually lost a base, right? This is no longer a codon. And so we can imagine that somewhere down the line there's no longer a codon there. And there's all sorts of stuff that could happen here, all sorts of problems that could arise. So here's an example that we've worked through. You have a regular RNA strand, It produces arginine, proline, arginine, glycine, alanine. If we perform an insertion, so here we randomly added an A, now we have a glutamine, an arginine, and a glycine, and then something else. We would actually need the rest of the DNA code or the mRNA code to fill that out. Certainly there are bases there probably, as we haven't hit the stop codon yet, but we don't know what they are yet. Meanwhile, if we formed a deletion, and so in this case we deleted the G, or what did we delete? We deleted something. GGG. Oh, well, I deleted something that is not here, I guess. Oh yeah, so I did delete the G, and so when I deleted this G, it brought those two C's together, and then the G's, and you can see that we end up still having a glycine, so it almost looks like there's no effect, but we don't know what kind of effect it has on this amino acid without more amino acids there, or more nucleic acids. So it has a pretty significant effect. And so that's pretty much the case of frame shifts. They're always going to have a pretty large effect on the overall overall code. Okay, I don't have much more to say about that, but we are going to talk a little bit briefly about chromosome or chromosomal mutations, which is a concept that we brought up at a previous time, but I want to just change our language a little bit, talk about it a little bit more clearly now that we know more about what DNA looks like. So these are the chromosome or chromosomal mutations. In my original text, they were labeled as large-scale mutations. And these include large-scale deletions, amplifications, translocations, and inversions. These are chromosome modifications that take likely a sequence that is already working or has some function and either deactivates it by taking out large sections of it through a deletion, or copying it, making more copies of the same protein. It could also end up being translocated to a different chromosome where that same protein would have a new function, and also inversions, which might reactivate or deactivate sequences of the DNA but keep the sequence whole. So a large-scale deletion, this could delete complete genes or multiple translatable sequences of the DNA, and that would lead to the loss of those genes within those regions. This might be very harmful, but it could also might not be harmful. Remember that if it's in an intron that's being deleted, then it might not have any effect at all. If it's in a exon, then we have a larger worry. So that is one. And the loss of information is also something to be worried about. Large-scale amplification and duplication is another option. So here we get a copy of the genetic sequence that's created, and so multiple copies of the gene could lead to the overproduction of proteins, similar to some of the effects of someone born with a trisomy, where they have extra copies of a single chromosome. That extra copy of a gene could create extra copies of that protein, and those extra proteins could interfere with function, or they might use up too many amino acids, which make it more difficult for other proteins to form. There's all sorts of issues that could happen there. There are also examples of the salivary amylase gene, which people who descend from cultures that have traditionally eaten starches tend to have extra copies of that salivary amylase enzyme sequence. And so that can improve the amount of sugar they can extract from starch. And this is known as evidence. This is used as evidence of coevolution between culture and DNA, I guess, or culture and traits. So they influence each other. I believe we talked about this in a previous section on evolution. Now, translocation is another option. Translocation is where a sequence from one chromosome is moved to another, and this movement of genetic sequence, we can see now how that might be very essential to function. Different chromosomes are open in different areas. Different locuses are open for different chromosomes in different types of cells. So an eye cell might open up, chromosome whatever, and let's just call it 5, and it might open up a location on chromosome 5. But in your skin cell, it opens up chromosome 6 more. So there's more genes on chromosome 6 active in your skin cell, and there's more genes active on chromosome 5 in your eye cell. But if you move a gene sequence from chromosome 5 to 6, then all of a sudden that gene will be more present in those skin cells, and that might have some interesting effect. Certainly, this is the explanation I've given before for snake venom that I've read about, at least, is that snake venom has a lot of similarities to a stomach enzyme that other snakes and reptiles have. And so the idea is that the enzyme sequence for the stomach enzyme sequence was translocated, maybe amplified and then translocated so that they were able to still have that enzyme in their own stomach. And that enzyme was translocated to a different chromosome, and that chromosome is more active in the mouth cells. And then over years of evolution and natural selection, that enzyme has become a better and better at breaking down and interfering with the function of living creatures. So it becomes a deadly venom. Now, an inversion is an interesting idea. An inversion is where a sequence is inverted so that the sequence of DNA no longer has the same function, even though the sequence has been preserved. Because the sequence has been flipped over, it no longer has the function it had before, since the order is inverted, but the sequence is preserved. So that's an interesting concept, because evolutionarily this would allow for a trait to disappear, or a protein to disappear in one organism, and then its one ancestral organism, and then reappear in an organism later on in the evolutionary chain. So we could kind of think that, in our cladogram or whatever, we might be able to say that, or in our phylogram, or phylogenetics, we could imagine having an example where we have organisms up here that have protein A, and then organisms here that don't have protein A, no protein A, but then their offspring have an inversion that allows them to have protein A again. So that's sort of an evolutionary idea there, that this shared organism back here had protein A, then it lost it, but then the offspring of this one were able to regain protein A by an inversion. So the sequence gets inverted. Somewhere along here, a mutation happens, inverts the sequence, deactivates this protein, and then later on, another inversion reactivates that protein. I don't have any immediate examples of this off the top of my head, but I am told this is something that we have seen evidence of or have used as ways to fill in some of the gaps in our phylogenetic trees, or explain away some of the something that might seem like an impossibility. So yeah, and we have observed this sort of action happening. These DNA strands are nice and loose and loopy, and so these loopy strands might get hit with an enzyme like a ligase, or one of these proteins like Cas9 that can actually cut the DNA sequence. It gets cut and then reassembled, but when it's repaired, the repair is actually at the wrong spot, and the DNA sequence ends up repaired instead of repairing as a loop, it gets repaired in a straight line. So some of these effects of these mutations might be a loss of function. So a mutation could result in the protein product that has little or no function. So by being short, the amino acid strand being shorter or longer, it no longer folds into the shape that can do the function it needs to do. You could also have a gain of function. So the new polypeptide that you're making, it might be a better polypeptide. It might be more effective. You can imagine very curly hair, for example, has a lot of cysteine residues. So simply having some mutations that create more cysteine residues gives you extra curly hair. Extra curly hair is really effective at, I believe, at insulating the body and other things like that, and repelling water. So those maybe, and so those examples. I'm just thinking about dogs right now. I believe that that things like poodles have extra repellent hair because maybe the curls trap oils. Now I'm just speculating, but there's a gain in function by simply replacing some of the amino acids with cysteine. You get extra curly proteins. You could also have lethal mutations. So of course a protein could become lethal in the body, and then that could create some of those examples of lethal genes that we discussed in our genetics unit. And finally, you could also have morphological mutations, ones that have an outward appearance change, but not really a change in function. So a change in color and shape. A lot of colors are based on the formation of various organic chemicals, which is all going to be mediated by proteins. If those proteins change a little bit, then the organic chemicals that make those pigments will be modified a little bit. And those little modifications can simply cause a change in the pigment, which may or may not have an impact on its evolutionary success. So some of the adaptive aspects of mutations, well, mutations, of course, are not always bad. They provide a source of genetic change in the biological world. We do associate mutations a lot with things like cancers, but there are beneficial ones that in the long term, of course, we're talking about thousands of generations term, would provide a benefit to the population through natural selection. Even the sickle cell anemia that we discussed earlier, that sickle cell anemia has some benefit to the population, and so it persists through the population. Here's an example of a bacteria that becomes resistant to an antibiotic by simply having the enzymes in its body modified. So, for example, one of the antibiotics, the one that's used to attack this Helicobacter pylori, the way the antibiotic works is that it is absorbed through the cell wall, and then the enzyme that it naturally produces converts the antibiotic into a poison. This way you can, if it's a bacteria that's in your body or in an organism's body, they can absorb an antibiotic that's not deadly to them, but it is deadly to the bacteria. But if a mutant arises and that mutant lacks the ability to produce that enzyme, so as we said, very common for mutations to simply stop a protein from forming or making it form in a less viable way. Now, without this protein, it can't digest the antibiotic. Without digesting the antibiotic, it does not turn that antibiotic into poison, and now this bacteria is able to survive. So it survives because of less information, and that's sort of a fun thing to wrap your head around. Okay, we're going to play around with some of this idea of mutation in the next section where we're going to talk about applications, but we are wrapping up on the end of this unit. The next couple sections are really idea-based and just interesting topics to talk about, so you'll just hear me ramble on for a little bit. But hopefully you're enjoying the material, and we'll talk to you soon.